So, welcome participants. Welcome to 19th February. Today is Shivaji Chhatrapati Jayanti. He was a great warrior from Maratha region, and I honor this day to him. May his soul will keep encouraging us for being a warrior and win over truth over the time. So on that note, so we were left with the blotting techniques from yesterday, from the Western blotting. So we continue with that. So we did with the southern, northern. I shared already this video, and the western blotting is done to detect your protein particle with the help of primary and secondary antibody. So here you can see that there's a nitrocellulose membrane here attached with the proteins. You put primary antibody, then secondary antibody, then you can visibilize these protein on the gel. So information obtained from western blotting is the size of protein that you have and expression amount of protein. So uses of western blotting could be used like it's mostly used as medical diagnostic technique, and a positive western blot can usually confirm an HIV infection. So in these things it is being used. So let's watch how it is done now.
In this video, you will learn how to perform a Western blot. A Western blot can be used to identify specific proteins in a sample and provide information about the protein size and relative abundance in the sample. First, fill a tray with blotting buffer. You'll be using this buffer to equilibrate your gel prior to starting the Western blot. Next, remove the gel from the gel cassette using the opening key. Line up the arrows on the opening lever with the 4 arrows on the cassette to open the cassette. After trimming the top and bottom of the gel with a straight edge, equilibrate the gel in the tray with blotting buffer for 15 minutes on a rocking platform. Pre-soak fiber pads in blotting buffer so that they are thoroughly soaked. To make a blotting sandwich, obtain a container large enough to fit the gel holder and add enough blotting buffer until the container is filled approximately 1 centimeter deep. Place the gel holder cassette in the container with the black side down and immersed in the buffer, and the white side up and out of the buffer. Lay 1 fiber pad flat on the black plastic. Next, wet a piece of blotting paper and place it on top of the pad. Be careful to avoid any bubbles between the pad and the paper, and make certain the buffer covers the paper. Take the gel and carefully place it squarely onto the blotting paper. Again, being careful to avoid any bubbles between the gel and the blotting paper.
Hello guys, welcome back.
Next, you will be applying a piece of nitrocellulose membrane. Remove the protective sheet from the membrane and wet the membrane with blotting buffer. Carefully place the membrane squarely on the gel. Avoid moving the membrane once placed on the gel, as proteins will begin to blot immediately. Using a roller, remove any air bubbles between the gel and the membrane. Place a second sheet of wet blotting paper on top of the nitrocellulose membrane. Place a second wet fiber pad on top of the blotting paper. Fold the clear plastic side of the gel holder over the sandwich and clamp it to the black plastic side by sliding over the white clip. This tight fit will squeeze the sandwich together. Insert the gel holder into the inner module. Make certain that the black side of the gel holder is next to the black side of the module. Place the inner module into the electrophoresis chamber. Add a frozen cooling unit and fill the chamber with blotting buffer to the level of the white clip on the gel holder. Place the lid on the electrophoresis chamber. Connect the electrical leads to the power supply, making sure the connections are correct, red to red and black to black. Turn on the power supply and run the blot at 20 volts. If a timer is available, set it for 2.5 hours. When the run is complete, turn off the power supply and disassemble the electrophoresis chamber and remove the inner module. Open the module and place it in a container filled with blotting buffer with the black side down. Starting with the first fiber pad, remove each layer until you reach the nitrocellulose membrane. As you remove the membrane, note that the proteins have been transferred from the gel to the membrane. Note that the kaleidoscope pre-stain standards have been transferred and can be seen on both sides of the membrane. You can also see that there are no longer any proteins on the gel. Immerse the membrane in 25 milliliters of blocking solution and incubate it for 15 minutes at room temperature on a rocking platform. Pour off the blocking solution. Add 10 milliliters of primary antibody. Incubate for 10 to 20 minutes on a rocking platform. The platform should be set at a faster setting to ensure constant coverage of the membrane. Pour off the primary antibody. Rinse the membrane quickly in 50 milliliters of wash buffer and then discard the wash buffer. Add another 50 milliliters of wash buffer to the membrane and let it wash for 3 minutes on the rocking platform at a medium speed setting. Discard the wash buffer. Add 10 milliliters of secondary antibody and incubate the membrane for 5 to 15 minutes on the rocking platform at a fast speed. Pour off the secondary antibody. Rinse the membrane quickly in 50 milliliters of wash buffer and discard the wash buffer. Add an additional 50 milliliters of wash buffer and wash the membrane for 3 minutes on the rocking platform on a medium speed setting. Discard the wash buffer. Add 10 milliliters of substrate. Incubate the membrane with the substrate for 10 to 30 minutes with either manual shaking or on a rocking platform. Watch for the color development. Once the colors have developed, rinse the membrane twice with distilled water and blot dry with a paper towel. Air dry for 3 to 60 minutes and then cover in plastic wrap for storage.
So that was about Western blotting. Good evening, बच्चों. Now let's go for now come the southern Western blotting. It will be mixture of both DNA and protein, how they both are interacting. In order to do so, we do the southern Western blotting. And northern Western is the RNA and protein interaction, how RNA and protein are being interacted, and we can detect on the gel with the help of hotspot. Whereas the dot blotting is done to identify and analyze the protein of interest. Methodology differs from the traditional Western blot techniques by not separating protein samples but using electrophoresis. So zoo blotting is based on the principles of southern blotting. And in the genome of any organism, there are two regions, the coding and non-coding regions. So it is the coding regions that causes the most of your research that you want to get associated. Just a second.
कल आपकी दावत है।
ओहो, क्या अंकल जी किसका है? क्या
कल सब शाम को दावत है
किस
खाने पीने की।
क्या है अंकल जी?
अरे ऐसे मतलब घर में पूजा पाठ।
ठीक है ठीक है ठीक है, चलो। So I was invited for a dinner party tomorrow. That's good. Uh, Now, yeah, so where we were in the southern, in the zoo plotting. So in this, in genome there are coding and non-coding regions. So researchers are more specific, interested with the coding regions. Um, so what we do in the zoo plotting is we distinguish between non-coding and coding region actually. So therefore DNA is first extracted from the related animals like human, monkey, mouse, and the sample is DNA. So zoo in each cut off the suitable restriction enzymes, and fragments are run on gel and transferred to a nylon membrane. And they are probed using DNA that is susceptible of being human coding DNA. So here you can see that zoo plotting of the rat, cow, dog, human. We have a loading slots. On the left side is non-coding regions that bind to only itself. On the right side is the coding regions. So all species have related sequence in that case. So in the zoo blotting technique, the probe of segment of human DNA may or may not be from the coding region, but blotting came out to be as usual. On the left, only the hybrid seen was between the probe and human DNA. Therefore, related sequence were not found in other species, and probe is probably non-coding. So that was it for the blotting point of view. So this presentation has been shared with you already. Mm. Mm-hmm. D8. So you will like this chapter. So here what we do in the fluorescence activated cell sorting, we usually sort between different cells having different properties like cancerous cells or normal healthy cells, and we want to sort them between healthy and non-healthy cells. In that case, we do the FACS analysis. We also call it FACS analysis. That is fluorescence activated cell sorting. So in this topic, we will cover the basics of flow cytometry, understand how flow cytometry sorter works, understand how cells are sorted, description of different cell sorting possibilities, tips on small preparations, and overview of sorting services provided by the FCF. So in this technique, you take your sample. So they could be fluid or tissue, and the fluids could be analyzed by FACS analysis, microfluid analysis, or large particle sorting COPAS. The tissue could be analyzed by the laser microdissection microscopy. So these are the various techniques we can separate our cells and identify them. So mainly, what is flow cytometry? Flow means fluid, cyto means cells, and metry means measurement. So there are types of particles that can be analyzed. So like cells, chromosomes, bacteria, yeast, microparticles. So on the left you can see different cells with various chromosomes, and yeah, bacteria, yeast, microparticles. So everything is possible with the help of FACS analysis. And we can analyze various sizes also, from bacteria of zero point 0.5 ranging to monocytes of 14 micrometer largest. So what does it measure? It's basically measures your relative size, relative internal complexity, how your internal cells are complex, and how the fluorescence intensity being absorbed by the cells. So based on these three main factors, that is your size, complexity, and intensity. Based on this, we work on the FACS analysis. So these are the various examples of sample analyzers. So like from the BD Bioscience, all from the BD Bioscience here, with their various capacities and various levels of working of them. So sorter, the FACS analysis in the sorter could be mainly of three subsystems it will have. It will have fluid department, optics department, and electronic department. In the fluid department, it will include focus on the stable break of sorting, that your cells are sorted out clearly and nicely. Whereas the optic part is generate the collection and collect the light signals. And electronics, it collects these optical signals proportional to digital signals and process to the computer, communicate with the computer. So how we record in this the fluidic part? So in the fluidic part, the sample is first introduced into the running sheet, that fluid in the flow cell. So here is your flow cell is going on the center core. On the side, it's hydrodermically focused in the core stream. Sheath, fluid, and sample do not mix in this case. And reduction of cross section causes an acceleration of the center core stream that leads to laminar fluidics. And sample flow rates are adjustable. So 10 microliter per minimum minute, 35 microliter per minute, 60 microliter per minute. So like this. So there are two different optic systems in a sorter. Excitations optics consisting of lasers, fiber optic cables that carry beam to steering prisms, which then direct laser beams to stream. So you have fiber optics, X-mount plate like this. So there are two different optic systems in a sorter. So collections optics consisting of fiber optic cables that directly emit the light to the appropriate emissions filter block, and filters that direct signals in the emission block to the appropriate photon multiplier tube. Here's your cuvette. From this, your fluorescent objective lens could be seen, and you have individual collection fibers. These are the various arrangements of optics, trigon and octagon. We will study about them in a video now soon. And this is a PMT, photon multiplier tube. So you have a cathode focusing electrodes here, and you put photon of light on this, and then vacuum tube with the diodes, and then they are being deflected like this with different diodes, and then absorbed at the anode side, and then you have current at the end. So how are cells actually sorted? So cells are sorted basically, you can see sample generates light scatter first, with fluorescence signals are analyzed, then the charge is applied by the stream charging wire, then deflection plates are attracted to repel each other. So your cells are going with charged droplets, and they break off based on the charge on the left and right. Neutral will go down, the one with the positive and negative, the red and green, they got deflected, and they get interchangeable at the end. So by this you can see the droplets get break off, and then according to the charge they are being separated. So into what collection devices can sort my samples? So you can use bulk sorts also, like 1.5 ml per tube, 5 ml FACS tube, 15 ml. So there are various tubes you can collect your samples. And then there are plate sorters, so you can use 6 to 384 well plates to sort your cells. There are slide sorters like microscopy slides, EBID, IBD type chamber slides, so all these things. So some quick tips to prepare samples. So plan experiments well first, that there should be optical configuration to be done, and unstained non-transfected cells to estimate the background signals, and bring FMOs if needs to set gates, and maximum of 2% of FCS BCA is recommended in the suspension medium of the cells to be sorted, because other cells might be sticky. You can also use following things to make it not for aggregation, that is, use calcium magnesium free sorting buffer or add EDTA. If the cell's preparation includes increased cell lysis, use 25 microgram per ml DNase 1 or 5 millimole MgCl2, and you can use 1% acutase in sorting buffer also. So all samples need to be filtered, preferably directly before sorting. So for a bulk sort, provides a cell with flowing concentration depending on the nozzle. So if there is 70 micrometer nozzle, so you can use 40 to 10⁻6 cells per ml. 85 micrometer nozzle, 20 to 10⁻6, like so on. So these are the nozzle size. You can use various work. So in summary, if you want to analyze your sorter, you can choose sorters you want to choose. Possibility to sort the smallest bacteria to largest adipocytes are possible. Plan your fluorochrome panel which you want to use for sorting. Make sure that you prepare samples following according to the guidelines. Do not forget to use controls and appropriate collection media for downstream technique. So that's was the basics, very basics about FACS analysis. So first, after your lecture is over, my first task is to share with you the slide, because I might today see the signals are so fast. Yesterday the file have to be uploaded. It took me like, so yesterday file size was, it was also big. It took me almost half an hour to be uploaded. But today, within seconds, but it was also small. So now let's see some interesting videos about it. FACS analysis, 2 videos I will play, 3 or 4 maybe, to understand it better.
Hello guys, in this video I'm going to speak about a very important or very interesting mechanism called the flow cytometer and FACS. Actually I did 2 videos, and this video I'm going to mention specifically flow cytometer, and in the next video I'm going to mention the FACS mechanism, fluorescence activated cell sorting. Let's start speaking about flow cytometer. Flow cytometer is a cell counting mechanism. So it's used to count the cells and to analyze the phenotype and the health situation of the cell. How does it work in general? First, in general, in flow cytometer, I have something called the flow cell. In the flow cell, we add our cell mixture. Then I have the sheath fluids. The sheath fluids will apply something called the hydrodynamic focusing. The hydrodynamic focusing of the sheath fluids will send the cells one by one through the flow cytometer tube. It's very important to pass the cells one by one through the tube, because here I have something called the laser beam, and the cells should pass through the laser beam one by one. This point where the cells pass through the laser beam is called the interrogation point. Now, when the cell pass through the laser beam, there are two events in every other direction, in side directions. These two events, or this light scattering, is detected by specific detectors called the forward detector and the side detectors. And these detectors are connected to a PC or a data analysis analyzer, which is going to analyze the data obtained from the detectors. Now, this is in general. Now, I'm going to speak more in details. Saying that this is the flow cytometer tube, and then the cells pass one by one through the laser beam. When the cell pass through the laser beam, the cells scatter the light in a forward direction, which is the light scattered in an acute angle. This forward scattered light is directly proportional to the size of the cell. Why? Because the cells scatter as like a particular amount of light in the forward direction, but when the cell is bigger, the forward scattered light will be larger, or like more forward scattered light will be detected. And when the cell is smaller, the cell will scatter less light in the forward direction. So the forward scattered light is directly proportional to the size of the cell. Now, the detector detects the pulse of photons, which are the forward scattered light is a pulse of photons, which are detected by the detector. And the detector then is, as I told you before, is connected to a PC or a computer. The PC will convert the intensity of the light detected by the detector into voltage. And then I will get a specific amount of voltage for every cell passing through the laser beam. The PC will give me a data similar to this. So every time a cell passes through the laser beam, I will get a specific peak, like high voltage peak for large cells and low voltage peaks for small cells. The data, or the forward scattered light data at the end, will be similar to this. Time versus voltage, and then the peak will be proportional to the size of the cell. This is forward scattered light. The second event happening in the flow cytometer is the side scattered light. So when a cell passes through the laser beam, the cell will scatter the light in side direction. And this side scattered light is directly proportional to the complexity and granularity of the cell. Why? Because the simple cells scatter the light in the side direction. Yes, but when we have granules inside the cell, so when the cell is complex from inside, these little granules will also scatter the light in different directions. And in this case, I will get more side scattered light, like this. So this is a complex cell from inside. It contains small granules inside, and then these granules will also scatter the light in different directions. And in this case, I will get higher intensity of the side scattered light. Similarly to what we saw in forward scattered light, the detectors are connected to the PC, which is going to convert the intensity of light into voltage, and then I'll get something similar to this. So the side scattered light data will be similar to the forward scattered light data. So for the complex cells will give me a high peak, and the simple cells will give me a small peak. Now, in order to analyze the cells, what I have to do is that I can combine both forward scattered light and side scattered light data together. In this case, I'll get something called the two-dimensional histogram. The two-dimensional histogram, from its name, it's two-dimensional. So I have side scatter and forward scatter data all together. In this case, what I get is something similar to this. So I have here 4 quadrants. If the cell appears in the lower quadrants, it means that it has low signal on the side scatter detectors, which means that the cell is simple. If the cell appears in the upper half, it means that it gives a high signal on the side scatter detectors, so it means that the cell is complex. If the cell appears on the right half, it means that the cell gives high signal on the forward scatter, which means that the cell is big. And if the cell appears here, it means that the cell is small. So if the cell appears, for example, in this quadrant, it means that the cell is small and it's simple. If the cell appears here, it means that the cell is small but it's complex. Here, it means that the cell is big and complex, and here, it means that the cell is big but simple. I will give you a very good example of this, which I have an example about the analysis of a sample of white blood cells. So what we see here is an analysis of white blood cells. This is side scatter and this is forward scatter light. What we see here is a group of cells which are complex, very complex cells, because they give a high signal on the side scatter detector, and they are medium inside. They are not so big and they are not small, so they are medium inside. Obviously, these cells are granulocytes, because granulocytes, or like eosinophil, basophil, neutrophils, they are medium inside, but they are very complex cells. Here we have bigger cells, because they give almost like they give more or less high signal on the forward scatter, but they give low signal on the side scatter. So they are big, but they are not complex. They are simple cells, and these are monocytes, because monocytes are big cells, but they are not complex. Here we have very simple cells, because they give very low signal on the side scatter, and they are like smaller than monocytes and granulocytes. So obviously these are lymphocytes, because the lymphocytes are smaller in size and they are simple cells. Of course, this is a very useful technique used to count the cells and to analyze the phenotype and the health situation of the cells. And what's very special about this technique is that we can analyze many thousands of particles and cells per second. It's like it's an easy technique. It's not very hard to perform, and it's precise. It's very used nowadays in all the laboratories. This is everything I wanted to tell you about flow cytometer, which is very important to know if you want to know more about FACS mechanism. In the next video, I will speak about FACS mechanism. So if you want to know more about FACS mechanism, then you need to watch the next video. If you like this video, please don't forget to like, share, subscribe to the channel, and if you have any questions, leave it in the comments. I will answer you, and see you in the next video. Bye.
So that was about the basics of FACS analysis. Now let's check how do we analyze this data. I hope our internet is working also. Is it so? Yes, it's working. That's good.
Hello guys, this is a continuation of the subject flow cytometer and FACS mechanism. In the previous video, I spoke in details about flow cytometer, and in this video, I'm going to speak about FACS mechanism, fluorescence activated cell sorting. If you didn't watch the previous video, I highly recommend you to go and watch it, because it will be much easier to understand FACS mechanism if you fully understand the flow cytometer mechanism. So let's start speaking about FACS. FACS mechanism is not only a cell counting mechanism, but it's also a cell sorting mechanism. So it provides a method to sort a heterogeneous mixture of biological cells. It depends on light scattering, as we saw in flow cytometer, so forward scattering in light and side scattering light, but it also depends on the fluorescence, and it also can detect fluorescence. FACS mechanism looks like this. So we have the machine, we have of course the computer, which is going to analyze the data, and we have the fluids container and the waste container. Now, how does it work? A FACS machine looks like this from inside. So we have also the flow cell and the sheath fluids, which apply the hydrodynamic forces on the cell to pass them one by one through the tube. This is what we saw in flow cytometer in the previous video. The cells will pass one by one through the cell tube, and then we have the laser. So the cells will pass through the laser beam one by one. When the cell pass through the laser beam, there is the forward scatter, which is detected by the forward scatter detector, and then there is the side scatter, which is detected by the side scatter detector. But here there is also the fluorescence, which is detected by the fluorescence detectors. Here we have mirrors and filters, which are aimed to filter the light and to deliver particular wavelength to the appropriate detector. Then all the detectors are connected to the computer, which is going to analyze the data. Now let's speak more in details. As we saw in in flow cytometer, we have forward scatter and light and side scatter. Here we have also the fluorescence. So saying that I have a cell mixture. Inside this mixture, I have a mixture of cells, or cells who are expressing a particular protein, and other cells who are not expressing the particular proteins, the particular protein or the protein of interest. What I want to do here is I want to count how many cells there is inside the mixture, and how many cells are expressing the protein and how many cells are not expressing the protein. And then I want to separate this heterogeneous mixture into two groups of cells. The cells who are expressing the protein and the cells who are not expressing the protein. To do this, first of all, I have to design a specific antibody for this protein. This antibody is specific to the protein of my interest. I can buy this protein. There are companies who design specific antibodies for particular proteins. Then I have to label this antibody with a specific fluorophore. Now, what I have to do is that I have to add this antibody, labeled antibody, to the mixture of cells I have. When the antibody is added to the cells, this antibody is going to bind specifically to the protein of my interest, and then I will have labeled cells. So I will have the cells who are expressing the protein will be labeled with this fluorophore. Now, when the cell passes through the laser beam, this is going to happen. First, I will get the scattered light, which is detected by the forward scatter detector, and this is proportional to the size, as we saw in the previous. We saw this in details in the previous video. So forward scattered light is directly proportional to the size of the cell. Side scattered light is directly proportional to the complexity of the cell. And then the fluorescence will give me an information about which one of the cells is expressing the protein and which one of the cells is not expressing the protein. Now, when the cell pass through the laser beam, this is the laser beam. First of all, this is the forward scattered, which is detected by the forward scatter detector. The side scatter, which is detected by the side scatter detectors. And then when the fluorophore pass through the laser beam, the fluorophore will be excited. And then when the fluorophore is excited, the fluorophore will emit light at a particular wavelength, and then this light will be detected by the fluorophore detectors. All these detectors are connected to the computer, which is going to give me the exact data about every event of these three. Now, of course, when the cell is expressing more proteins, there will be more fluorescence coming out of this cell. At the end, I will get a data similar to this. So when the cell is expressing the protein, I will get a high peak, and then when the cell is not expressing the protein, I will get a small peak. And then the computer will give me something called the one-dimensional histogram, which is something similar to this. Here we have the cell count, or the events. We call them the events passing through the laser beam. And here we have the signal of the fluorophore. So when I have a high signal of fluorophore, then these cells are the cells who are expressing the protein of interest. And the cells who give low signal on the fluorescence detectors are the cells who are not expressing the protein. What I can do further is to combine this data with other data I have. So for example, I can combine the data obtained from the fluorescence detectors with the data obtained from the forward scatter detectors, for example. So here I have the fluorophore, here I have the forward scatter, or here I have the fluorescence and forward scattered light. When the cell appear here, it means that these cells are small cells because they have low signal on the forward scatter and they have low signal on the fluorescence detectors, so they are not expressing the protein. When the cell appear here, it means that the cell is not expressing the protein because it has low fluorescence, but it's a big cell because it has high signal on the forward scattered light. Then when the cell appear here, it means that it's expressing the protein, but it's a small cell. And when the cell appear on the upper right quadrant, it means that the cell is big and it's expressing the protein. So this is what we call the two-dimensional histogram. I can combine two data together and then I get something like this. Now, what's special about FACS mechanism, or why it's called FACS? FACS means fluorescence activated cell sorting, which means that this mechanism cannot only give me a cell count, cannot give me a data about the cell count or the percentage of the cell expressing the protein and not expressing the protein, but also the FACS can separate the cell mixture into two groups of cells. How does it work? Let's see. This is the FACS mechanism. So here we have the flow cell, and here we have the cells passing one by one through the laser beam. When the cells pass one by one through the laser beam, as we saw before, it will scatter light forward and side scattered light, and also fluorescence. All these events will be detected by the detector. At the end of the tube, there is something called drop formation. In drop formation, we have a mechanism called a vibrating mechanism. This vibrating mechanism will separate my mixture into small droplets. These droplets should contain only one cell by droplet. This is very important. Then what I can do as a user is that I can adjust the settings of the program in order to charge the cells. So at the end of the tube, I have something called the electrical charging ring. The electrical charging ring is responsible to charge the cells according to my adjustment. So, for example, if I adjust the settings, for example, of the program, I can ask the computer to charge the high fluorescence cells with a positive charge and the low fluorescence cells with a negative charge. Then the electrical charging ring will charge the the cells will pass through the laser beam. The detector will detect the fluorescence, and then the computer will charge the high fluorescence cell with positive charge, for example, and the low fluorescence cells with negative charge. I can choose, actually. For example, if I have this data, this is the two-dimensional histogram, I can choose to charge the big high fluorescence cells with a positive charge, and the big low fluorescence cells with a negative charge, and the other cells with no charge. Then the neutral cells will go to the waste. These cells might be like debris. These might be like just debris. I'm not interested in them, so it depends what cells I'm interested in. So I can adjust the settings of the program, and then the electrical charging ring will charge the cells. Then I have different electrodes, like negative and positive electrodes, and then the positive cells will go to the negative electrodes, and the negative cells will go to the positive electrodes, and the neutral cells will go to the waste. In this case, I will have two groups of cells: the cells who are expressing the protein and the cells who are not expressing the protein. And this is what we call cell sorting. Of course, I can also study two proteins, not only one. And this is done by using different... of course, to study two proteins, I need two specific antibodies, because I need one specific antibody for each protein. But I need to label every antibody with a different fluorophore. Of course, I have different fluorophores, and every fluorophore emits light in a different wavelength. I need to know every fluorophore, the wavelength every fluorophore emits light in. So if I use two different fluorophores, then I can study two different proteins. For example, this fluorophore emits a green light or a light green light, and this fluorophore emits yellow light. So they are different wavelengths. Then if the cell is emitting only has only a green fluorophore, we can know that this cell is expressing one protein, or the green protein. If the cell is emitting yellow light, then the cell is expressing only the second protein. And if the cell is emitting two lights, or giving on the two detectors, then we can say that the cell is expressing both proteins. How can we detect two different wavelengths? As I showed you before, the FACS mechanism contains different detectors, different fluorescence detectors, and here we have the filters, the light filters, which deliver particular wavelength to a particular detector. So every detector will detect a particular wavelength, and then we can use two different fluorophores if they emit light on different wavelengths. Okay. We can also combine the data together. So I can form this two-dimensional histogram again, but with the protein 1 and protein 2. So I have fluorophore 1 signal coming from fluorophore 1 and fluorophore 2. So when the cells are when the cells are here, it means that these cells are expressing both proteins, because they are giving high signal on the first detector on the second detector. If the cells are here, it means that they are expressing protein 2, but they are not expressing protein 1. These cells are expressing protein 1 because they have high signal on the detector of fluorophore 1, but they are not expressing protein 2 because they have low signal of on the detector of fluorophore 2. And these cells are not expressing any of the proteins. Now I have like a real two-dimensional histogram. And this is like ideal to see how cells are shown in the two-dimensional histogram. So these cells, here we are studying two different proteins, CD14 and CD4. We are labeling CD14 with PE, and we are labeling CD4 with FeedC. We are labeling them with different fluorophores. Then cells in this quadrum are expressing CD14, but they are not expressing CD4, so we have CD4 negative. Cells here are expressing both. They are giving high signal on both of them. So the CD14 positive, CD4 positive. These cells are CD14 and CD4 negative because they are giving low signal on both detectors. And these cells are expressing CD4. They are giving high signal on CD4 detector or on FeedC detector, but they are giving low signal on the other detectors, so they are expressing only CD4. This is like a very simple example about how the data looks like in the two-dimensional histogram. Of course, there are many more complex data. So if you want me, I cannot speak about everything in this video. So if you want me to do another video especially about data analysis and gating in facts analysis, please write me in the comments. As you know, now we can in the new fax machines we can analyze like tens of proteins and tens of cells in one time, because new fax machines have like some machines like there is now one fax machine in the market which contains 10 lasers and 30 detectors. Can you imagine 10 lasers and 30 detectors? So you can analyze many proteins and many cells at one time. But of course, the data then will be much more complex than this data. This is a very simple data. If you want me to talk more about this, then write me in the comments. If you enjoyed the video, don't hesitate to like the video, share, subscribe to the channel. If you have any questions or any suggestions for other topics, write me in the comments, and see you in the next video. Bye bye.
So that was all analysis related to that. And on that note, I think we are done for today. Abhay Srivastav. Does the direction of light scattered by the cell correlates to determine the cell size? Yeah, that's that they are proportional to each other, so they are being dependent. So I hope so we will start with that tomorrow. You might have the next lectures. If we have been on time, also I could have shared the another two videos. I think we will share them later with you. So that's it for today. We will do the rest of the things tomorrow. Thank you very much. I think there is some error being there. I cannot see my own video. So take care. Bye bye. See you tomorrow then. Yeah, I know. So basically, I have to just let you know how your different cell size, different characteristics, how you can differentiate between them according to their structural analysis, according to the cell size, how you can differentiate on the 2D histogram based on the FACS analysis, how it works and how things are working. So do you want to have class tomorrow also? I mean, or do you want break now? I, wa- or shall we take off tomorrow? Please let me know your views on that. So according to that, we will work. If you want class tomorrow, we can have a class because your 30 or few students are coming up. So we can manage one class for sure for you, all of you. Because if we have a class tomorrow and we can finish it on which platform, on which lecture. So we have bioinformatics then and scientific practices. We have 2 days work. We can finish it off by Sunday if you want, or we can finish it by Tuesday. Just let me know how it works in the group. So according to that, we will work. Yeah. On Monday. Okay, fine, fine. So tomorrow, no work. Okay, fine, fine. So we see us on Monday. Thank you. Bye bye. We continue with the facts analysis. Take care. Bye bye. Good night. Sweet dreams.